So this is the second part of our consideration of how neurotransmitters get packaged into vesicles and how the cell transports them from the cytosol into the synaptic vesicles. It's very easy to think of synaptic vesicles as a unique category, but they are part of a secretory process that is present in lots of different cells. And here we have a diagram showing the relative sizes of synaptic vesicles and the vesicles in larger cells that secrete hormone, here chromaffin granules from adrenal chromaffin cells. And the difference in size here is very important when one thinks about the capacity of these granules to store contents, either neurotransmitter or hormone to be secreted. In fact, when the neurotoxin MPTP was first discovered as a compound that could kill dopaminergic neurons and produce Parkinsonian-like symptoms in young adults who were nowhere near Parkinsonian age, it became highly studied as a neurotoxin. And one of the observations was that when it was applied to adrenal chromaffin cells, which have these large granules that are not only secrete adrenaline, but adrenaline's, epinephrine, it's a neurotransmitter. Those adrenal chromaffin cells were not killed off by MPTP. And that was a major surprise. But the result was that the monoamine transporter resident in the chromaffin granules was such a good transporter it could remove the applied MPTP from the cytosol and store it or sequester it inside the chromaffin granules, and the capacity was so large that essentially the MPTP was sequestered and therefore detoxified. So one has to think about the process and the function of these vesicles, not only from a secretory one, but also perhaps from a neuroprotective point of view. And what do we know about the composition of synaptic vesicles? What are they like inside? Is their environment the same as every other cellular environment we talk about? This is very hard to do since they're so small, and early biochemical studies examined the adrenal chromaffin granules because they were large and accessible and you could purify large quantities of them. So this is a table from a review in the 1980s that gives us some very interesting ideas. If you look at the protein lists that they were able to compile, we did not have proteomics at that point in time, so they could determine that 42% of the composition of the granules was indeed protein, but individual proteins were not exceptionally abundant except for the ATPase, which we know is what will acidify the granule environment, internal environment. The ionic composition, however, is much more interesting. And if you look over in the table on the right, we're looking at molar composition, or concentrations here. And the first ones let's go and look at are those of the catecholamines. So here we have adrenaline or epinephrine present at 360 millimolar. Well, now that's not so outrageous considering the concentration of serotonin we just saw measured in the secretory granules, large and small synaptic vesicles from the leech nervous system. What's surprising is not only is there adrenaline, but there's noradrenaline and dopamine, the two precursors for adrenaline synthesis. So the monoamine transporter, as a non-specific transporter, is bringing in all of the different monoamines. There is some selectivity here, and clearly maybe even some of the synthetic enzymes, so that any dopamine that gets in might be converted to the norepi, and then the norepi subsequently converted to the epinephrine. All right, so let's look next on the list at nucleotides. ATP, 87 millimolar. Cytosolic ATP is maybe 10 to 12 millimolar. This is a whopping concentration of ATP. What might so much ATP be doing inside a vesicle? Any ideas? Well, we now know that ATP is actually secreted as a neurotransmitter. There are purinergic receptors in postsynaptic sites, so that ATP is a signaling molecule as well as an energy source. And clearly there are processes to concentrate ATP inside the granules, secretory granules. All right, next off, let's look at the hydrogen ion concentration. 10 to the -5th molar. That's a pH of 5. So that's awful acidic. Cytosolic pH is 7.1. Extracellular pH is 7.4. So very special intracellular environment inside these chromaffin granules. Let's look at the next line. Calcium concentration, 14 millimolar. Extracellular calcium, 2 millimolar. Intracellular calcium, 100 nanomolar. This is a whopping concentration of calcium, and we have no clear idea how it gets there or why it is so high. Magnesium is also high, but only about twice as high as cytosolic. Look at the sodium and potassium. Sodium concentration 6 millimolar. That's not outrageously different from a cytosolic concentrate 10 millimolar, a little bit less. But potassium, intracellular potassium's 120, 140 millimolar. Here it's tenfold less, 12 millimolar. Very different composition of this intravesicular space in terms of ions. Very little osmotic pressure from mono and divalent cations. We have a milliosmolarity inside the cell of 300, 310 milliosmoles. Inside vesicles, that osmotic is not coming from the cations themselves. It's clearly coming from the neurotransmitters. All right, so what about the proteins? On the left, we have a 1980s view of the proteins that were identifiable in the chromaffin granule membrane, and on the right, we have a recent 2006 paper that actually quantitatively analyzed synaptic vesicle proteins to determine how many copies of each protein were present on average in synaptic vesicles. So this is a much more detailed look. Let's examine these. First of all, in the 1980s view, we see that we have the vesicular ATPase, which utilizes the energy of the ATP molecule to pump hydrogen ions into the vesicle. Well, that certainly accounts for the very high intracellular pH. There's also been identified the vesicular monoamine transporter molecule, which will be a transport pathway for the monoamines. There's a vesicular ATP translocase, which is going to concentrate the ATP inside the vesicle. This is a relative of the ATP adenine nucleotide translocator of mitochondria, which moves ATP and ADP across the mitochondrial membrane. But we always also see a cytochrome, which means that there's some redox reactions going on, probably to keep the monoamines in the appropriate redox state. And we also see some dopamine beta hydroxylase, which is the synthetic enzyme that takes dopamine and makes it into norepinephrine. So there is machinery in at least the granule cells, chromaffin granule vesicles themselves, to synthesize and maintain the epinephrine as epinephrine. In our more contemporary view, let's look for some familiar proteins. All right, first off, we see up here at the top synaptobrevin. That's our vesicular snare, and it is the most abundant protein in synaptic vesicles, and there's 70 copies approximately per vesicle. That's pretty impressive when one considers that 20% of the synaptic vesicle membrane is occupied by protein transmembrane domains. More of the surface area gets occupied by the bulbous lay of proteins, as you can see from many of these bulbous ones. All right, so what's the next one? Synaptotagmin, again up here at the top. Here's our calcium sensor, and you can see multiple copies of it in various places on this diagram. And here's synapsin in purple on the right. Synapsin is the protein that attaches the synaptic vesicle to actin filaments for storage inside the cytosol. But wait a minute, we've got MUNC18, SNAP25, and syntaxin also identifiable and quantifiable in synaptic vesicle proteins. Where are these supposed to be? You're right, they're supposed to be on the target membrane. These are not supposed to be on vesicular membrane. So what does that mean? That means that the sorting process, whether it's just in recycling as kiss and run or through the endosomes, is not perfect in order to keep synaptic vesicle proteins in vesicles and plasma membrane proteins in plasma membrane. And there's other examples of this kind of mislocation occurring naturally, which tells us that all biological processes are not perfect. All right, so what's this great big one? This again is our vesicular ATPase as it had been identified biochemically. And there's only 2 copies of that per vesicle, but it works hard. And then what other kinds of transporters do we have? All right, so the chromaffin granule, the monoamine transporter. Here in the central nervous system, synaptic vesicles, we've got the glutamate transporter as a small globule. We have a chloride transporter. So active chloride input, consistent with our idea that we need to bring chloride in to the cytosol to balance the hydrogen ion concentration. And then there's some other identified transporters also present. In the cutaway of the inside of a synaptic vesicle, I also want to call your attention to this meshwork of what turns out to be heparan sulfate proteoglycans and sialic acids and other glycosylation agents on the vesicular or luminal side of many of these synaptic vesicle proteins. And we'll come to what those might be doing in another slide or so. Right, this table comes from the same paper that did the molecular neuroanatomy, just to give you some idea of the expression numbers in copies of proteins per vesicle that came from that study.